Hello everyone. We are going to have a brief PowerPoint on pharmacogenetics. This is part 1, terms and meaning. This should be about 10 or 12 minutes, and then there will be a part 2. This is from chapter 10 in the book. There are some other embellishments that I'm adding, but reading chapter 10 after you view this will be really helpful. So what we're talking about here is how inherited differences in genes impact the therapeutics in your patients, specifically how drug metabolism and action is impacted by genetics. So there's two terms. There's pharmacogenetics and genomics. Really, they're interchangeable, although the genetics typically refers to just one or two genes, where genomics is the whole genome. For the purposes of this, I will probably use the terms interchangeably. The benefits of understanding this is because knowing pharmacogenetics ahead of time helps you predict drug reactions, so safety and adherence will be better for your patient. Specifically, we're talking about genetics variants in the cytochrome P450 family, which is our drug metabolizing enzyme superfamily, and this is your pharmacokinetics. It's particularly your metabolism. It can also affect your pharmacodynamics. It can affect how the receptors act, it can affect the numbers of receptors, and it can also affect things like drug transport change, where the drug is transported from the outside of the cell into the cell. The terms and definitions, I have an image which will really illustrate this much better, but these are the things we're going to look at. I'd like to just point out that genes code for proteins, and the CYP enzymes are proteins. So the genes are coding for the proteins, and we're talking about variants in the genes. We're going to talk about all the things that you see listed here. On this slide, what you see is how our chromosomes, if you start up at the upper left, it unwinds, and then you get all the way down to the lower portion, and that is a segment of the DNA, which is called a gene, and the arrow on the far right is pointing to that gene. The arrow on the far left is pointing to exon, and an exon is the area that actually codes, and that's what the mRNA actually transcribes. And as you'll see, different enzymes have different numbers of exons. The arrow below is pointing to an intron, and the intron is a non-coding area, and that also is cleaned up by the mRNA in the process of transcriptions. Just to file away the fact that genes code for proteins. Proteins are made up of introns and exons, which are then transcribed into proteins. This slide shows us the picture of an allele, and the allele is the little light blue segment in the chromosome, and we inherit one from each parent. An allele is a version of a gene, and so if we get the capital A and the small a, we've got two different versions of an allele, and that means that we are heterozygous. Whereas if we have two capital A's, we are homozygous. The type of allele of interest for this conversation has to do with the alleles of the genes that code for the cytochrome P450 enzyme. And we're going to talk a lot more about that. Our next topic is polymorphism, which is genetic speak for diversity. And it can be one thing that you might hear called a SNP, SNP, which stands for single nucleotide polymorphism. That's where just one of the nucleotides is changed. This is a way of talking about genomic diversity. It can be a loss of function, a gain of function, or another alteration in function. It's where the DNA sequence has one or two variations, and it must occur in at least 1% of people to be kind of repeatedly seen, to be referred to as a polymorphism. And you see examples here. It could simply be a C instead of a T, or a whole stretch of DNA that is present or absent. This is an illustration for you. The top two Variations shown are single nucleotide polymorphisms, just a single nucleotide, and then the bottom two are showing kind of a repeated version. So these are all polymorphisms. Another common genetic change or alteration in pharmacogenetics is in the area of the promoter gene. And the promoter gene is a really cool term to me because it's the actual area that kind of signals to the messenger RNA to come over here. This is the beginning of the gene. So it kind of directs the RNA to find that gene when it's time for transcription. In this picture, this depicts some of the possibilities that can happen with pharmacogenetics. So the little bunch of people up at the top are all people with the same diagnosis, and you're going to give them all lisinopril, we'll just say. So then if you go down to the lower left corner, just because of their type of pharmacogenetics, some people will have no benefit, but they will have no toxicity either. You're glad they didn't get toxic, but you wish they had gotten some benefit. Moving over to the right, we have people who have some benefit, but no toxicity. This is where you want to be. Moving up then to the group of yellow people, you've got the benefit plus toxicity. Then up at the upper corner in the right, you've got no benefit and toxicity, which is definitely not where you want to be. This slide is showing our cytochrome P450 enzyme families. The 6 primary families that we are interested in this course are the 3A4, which 47% of all drugs need in order to be metabolized by the liver. 2D6 is also very common with 25%, and then you can see by the little hat symbol there, there's lots of genetic variability with 2D6. There is cytochrome P2C9. 13% of all drugs require this to be metabolized. There is a lot of genetic variability there. And then the next one is 1A2, 2E1, and 2C19. Together, those 3 are metabolizing about 15% of drugs. So those are not as common, but 2C19 has a lot of genetic variability. Another visual representation of the CYP enzymes, they're in the liver, they're also in the gut. The pie chart on the left shows the approximate content of the CYP enzymes in the liver. The pie chart on the right shows the percentage of drugs that are metabolized by those CYP enzymes. So it's another different way of looking at it. The numbers might be slightly different than the previous slide. This slide shows the individual variation with the same drug. It's kind of a more graphic illustration of the previous slide that had the groups of people. So you see we've got 3 curves here, and obviously where we want our patient to be is in the gray area. They have the therapeutic range with no toxicity. That's the sweet spot. The lower range, that's your ultra-rapid metabolizers, ultra-rapid. So their liver has a lot of the enzyme. They're fast metabolizers. They take it. It does peak, but it's barely into the therapeutic range, and then their body's going to clear it. Whereas at the top, they're slow metabolizers. They've got that certain type of polymorphism so that the drug is hanging around in their body much longer. They are going to be more toxic than getting any benefit. Right now, we're going to talk about the lingo that's out there for the type of metabolizers. I want to start with the slide in this top column that's on the far right where it says normal. So normal metabolizers are what the people have, the type of CYP profile that have benefit with the therapeutics without the toxicity. So there's other names that I've seen in the literature. Extensive is also synonymous with normal, or wild type. I just point that out because you may see that. So if you see wild type, that is normal. And if you see extensive, that is also another word for normal. It's a little bit confusing, but I just wanted to make that point initially for you. Then at the upper left, we have poor metabolizers. So those are going to be the ones at risk of toxicity. Their body has inherited alleles that just don't code for a lot of the CYP gene that's needed, the CYP enzyme rather. Then intermediate, those are going to be a heterozygote. They're going to have one normal and then one different one that's a little bit slower. Moving down to the lower right corner, you have the rapid metabolizer. Those people have many copies of the gene, or they have a variant in the promoter gene. Remember, the promoter gene is a segment of the gene where the RNA kind of docks, where it kind of looks for. Then the last one are the ultra-rapid metabolizers. So again, they have many copies or a variant in the promoter gene. We're going to talk about the nomenclature now in pharmacogenetic testing. As you know, the allele is a version of the gene, and we inherit two alleles of each gene. So in terms of pharmacogenetics, what they call it is the star allele. So if you remember that picture of the chromosome with the light blue band, that would be a star allele. And it's given the term star 1, star 2, or star 17. So a person can carry two normal copies, star 1, star 1. Remember, normal is sometimes called extensive or wild type. A person can inherit one normal copy with a polymorphic, which would be star 1, star 2, or both copies could have a polymorphism, star 2, star 3. When it comes to the 2C9 gene, there are two main types found, two main types of SNPs or polymorphisms or variants. First, they have a normal enzyme activity, which is obviously not a variant, but then there's the 2C9 star 2. Those carriers of that polymorphism have a 30% decrease in activity. The 2C9 star 3, those people have as much as a 90% decrease in their enzyme activity. So think about if they have a decrease in enzyme activity, that's impacting the metabolism, or the blood level is going to stay high. So if a person has star 1, star 2, what type of metabolizer will they be? Where will they be on that slide that had the three curves? Or if they have star 2, star 3. This is another way of depicting the difference. You see it in a pie chart. They've got four types of metabolizers here. Remember that blue chunk where they're calling it extensive, those are the normal ones. So they have the star 1, star 1 alleles. The intermediate, there's 18 to 45% of those. Those could be star 1, star 2, or star 1, star 3. So they are going to have a normal allele paired with one that's going to have slower, decreased activity. And then the poor metabolizers are seen, as well as the ultra-rapid. Now we're going to talk about those in a moment as well. This last slide in part 1 has to do with something called the star 17 allele. So we've talked about star 1, star 2, star 3, and now we're going to talk about star 17. This is called the gain of function mutation, which makes the enzymes much more active. It's kind of the ultra metabolizer allele. So those are the ones, if you think back to our curve slide with the three curves, those are the ones who are barely going to make it up to the therapeutic range, and then it's going to be cleared because the enzymes are so much more active in clearing out that drug. So they're the ones who are going to have no benefit but no toxicity. I already answered the question, so think about that one.